Hello everyone, welcome to Physics Wala English YouTube channel. I'm Shalini Somashekar, your botany teacher, and in today's session, we are going to continue with the chapter Anatomy of Flowering Plants. In the previous class, we started with this chapter and we learned about the different types of plant tissues, and we also learned about the different tissue systems that we see in plants. So in today's class, we are going to take the knowledge from our previous class and apply it. Okay, so what exactly we are going to do in today's classes? We are going to learn about where exactly the plant tissues that we studied about in the previous class, how they are arranged in the different plant organs like the root, stem and leaves. If you have to find out how different tissues are arranged in these plant organs, you will have to look inside them, right? So in order to understand what are the different types of plant tissues, how they are located inside root, stem and leaf, we'll have to cut it open. So what we're going to do is we take extremely thin sections, extremely thin slices of the different plant parts, look at it, observe it under the microscope and find out what are the different tissues that you can find in plants. Okay, so in today's session, we're going to start off by learning about the transverse section of dicot as well as monocot root. What do I mean by TS here? TS here is a transverse section. So if you can think of this as a root, when you take an extremely thin section of the root, horizontal axis like this, then the section that we get is called the transverse section. So we're going to look at the transverse section of a typical dicot and a monocot root. Following that, we're going to look at the transverse section of a dicot stem and a monocot stem. And then finally, we're going to learn about the transverse section of a dicot root as well as a dicot leaf as well as a monocot leaf. And finally, we're going to learn about secondary growth. All right. So without any further delay, let's get today's session started. Here is a section of a typical dicotyledonous root. So let me first tell you how we arrive at this diagram. I'm sure all of you must have come across this diagram in your NCERT textbooks. So how we got this image is from the transverse section of a root. If you can think of this as a plant, this is the shoot system, this is the root. So if you take a thin transverse section of the root, you get a section that has a circular outline, right? So in the circular outline, outermost you have the epidermal tissue system. Inside you have xylem and phloem, all of that, right? So xylem and phloem will be arranged inside. In between you have the ground tissue. So when you take a pizza section like this, that is what is represented here. So what you're looking at here is one section of the transverse section of the root. So we are going to learn about what are the different tissues that are present in this particular section. The same thing applies to the rest of the part. Because it's difficult to draw it entirely, this is how it is shown to you. But whatever is existing here, what are the different features that we will learn about here, the different types of tissues that are present here, the same applies to the entire section. Okay, now let's start by talking about the different tissues that we see in the TS of a dicot root. We already learned in the previous class that epidermal tissue is the outermost covering for all plant parts, right? That applies for root as well. So in dicot roots also, the outermost layer is the epidermis. Here you can see in the epidermis, there are cells that are compactly arranged next to each other. The epidermal cells are parenchymatous in nature. They are made up of parenchyma. It is the outermost layer. The epidermis that we find in roots is also called as the epiblema. As you can see, some of the cells give out very thin extensions on the outer surface. These are nothing but the root hair. One thing we must remember about the root hair is that root hair are superficial and they arise from the outermost layer of cells in roots, which is the epidermis, and these root hair are unicellular. I don't know if you remember or not. In the previous class, when I told you about the tissue systems, I mentioned very clearly that epidermal root hair are unicellular, right? So the root hair that arise from the epidermis are unicellular. Now, when you have to talk about the function of these root hair, you already know from the chapter Morphology of Flowering Plants, root hair help in increasing the surface area. Now, what is the function of the root? Root has to absorb water and minerals from the soil, right? So, the presence of these root hair in the epidermis will increase the surface area so that more water and more minerals can more efficiently be absorbed from the soil. Okay, so that's about the epidermis. Also, what I want you to notice at this point is that there are no intercellular spaces in the epidermis. The reason is because it's protective in function, right? So it has to protect whatever is lying inside it. If there are gaps and spaces in there, protection will not be very effective, right? Therefore, the outermost layer, epiblema, has barrel-shaped cells, parenchymatous cells that are very closely arranged with very little or no intercellular spaces. And it gives out extensions that are called root hair, which increases the surface area that is available for absorption of water and minerals. All right, so that's about the epidermis. In the epidermis of roots, you do not find cuticle or stomata, okay? Because it's an underground part of the plant and it's not required there. Now let's talk about the region that follows the epidermis. Just below the epidermis, you have several layers of parenchyma cells. As you can see here, I'm going to outline them one by one. Let me use black. So you can see all of these cells here. These are also parenchymatous cells. Okay, so this region below the epidermis, which has many layers of parenchymatous cells which are thin-walled, that portion of the dicot root is referred to as the cortex. Though epidermis and cortex are both parenchymatous, you can see how their shape varies. These are more barrel-shaped and these are more spherical in shape. Another difference is, in the epidermis, you don't come across a lot of intercellular spaces, whereas in the cortex, you can see the presence of intercellular spaces. Okay, so epidermis is parenchymatous, single-layered. Cortex is parenchymatous, but it has several layers. All right, now let's talk about endodermis. Endo means what? Epidermis. Epi means above. It is the outermost layer, so that is the epidermis. Endo means inside. Endodermis is basically the innermost layer of the cortex. So here, it is the innermost layer of the cortex. It is a single layer. The endodermis also has barrel-shaped cells. See, this is the endodermis. Even here, you will have barrel-shaped cells. And they are also parenchymatous. Even here, you do not find any intercellular spaces. In the epidermis and the endodermis, the cell shape is pretty much the same. Though we say that endodermis is the innermost layer of the cortex, the cells of the endodermis resemble the epidermal cells more than the cortical cells. Okay, so in the endodermis, you will find a single layer of parenchymatous cells that are barrel-shaped and has thin walls without any intercellular spaces. There's one thing that is characteristic of endodermis in roots. I'll tell you what that is. If you think of this as a circle, this would be the radius. And this would be the tangent, right? Now, consider this particular endodermal cell. If you consider this endodermal cell, this will be its radial wall, and this would be its tangential wall. This is the radius, this is the tangent. So this will be the radial wall, this will be the tangential wall. In endodermal cells of the root specifically, in the radial as well as the tangential walls, you can find deposition of a substance called suberin. This suberin is impervious to water. It will not allow water to pass through it. It will not allow water to be transported through it. Okay, so there's a deposition of a water impermeable material called suberin, which is waxy in nature. Those are referred to as Casparian strips. This is very, very important, and you have to remember, I repeat it. Endodermis is the innermost layer of the cortex in roots. It has parenchymatous cells that are barrel-shaped. It is a single layer. no intercellular spaces. It has Casparian strips. Now, what are these Casparian strips? Casparian strips are nothing but the thickenings that you find in the radial as well as tangential walls of endodermal cells in the root. What do these thickenings do? Basically, they are impervious to water. They will not allow water to enter through it. Okay, so it basically acts like a checkpoint and make sure it kind of controls what enters the root vascular bundles and what shouldn't. More about it you will learn in a chapter called Transport in Plants. Okay, so now just remember there is something called Casparian strips that is present in the endodermis of roots, which is made up of suberin, which is a water impermeable waxy material. Okay, so this much is sufficient for you to know in this chapter. Okay, that's about the endodermis. Now let's talk about the pericycle. Pericycle is the layer of cells that is found below the endodermis. Okay, so in roots, it may be a few layers in thickness. Right here, below the endodermis, you can find the pericycle. It can be a few layers in thickness. Again, these are parenchymatous. Now, from the pericycle, two very important things take place. The initiation of lateral roots and the initiation of vascular cambium. Okay? So, what happens here is, in dicot roots, you know that in dicots there is a taproot system, right? So, in a taproot system, you will have the primary root. Branching from the primary root, you will have the secondary root. From the secondary root, you will have tertiary root. So, whenever a branch has to form from the primary root, the initiation of that lateral branch happens from the pericycle. Okay, so initiation of lateral roots takes place in the cells of the pericycle. Also, initiation of vascular cambium during secondary growth takes place in these cells. Now, what exactly vascular cambium is, what is secondary growth, you'll learn in detail in a few minutes in today's lecture itself. But I'll briefly tell you for now. Secondary growth basically refers to the increase in the girth of the plant. When a small stemmed plant grows, increases in girth and becomes and forms a huge trunk, the increase in girth of the plant or the diameter of the plant is what we call secondary growth. And for secondary growth to happen, cambium is responsible. A type of cambium called vascular cambium. The initiation of vascular cambium in some plants happens from the cells of the pericycle. That is what it means here. Okay. Now, these are two important things that you have to remember about the pericycle. At this point, I want you to notice how most of the cells in a root are parenchymatous. Epidermis is parenchyma, cortex is parenchyma, endodermis is parenchyma, pericycle also is parenchyma. Though they are all parenchymatous, we do see some variations among them, right? The parenchyma here is thick-walled. The shape of the epidermal parenchyma and the shape of the cortex parenchyma is variable. So, though they are all parenchymatous, you see some variation among them. Also, this confirms the fact that parenchyma is indeed the most abundant tissue that you find in plants, right? Now, let's move on and talk about vascular bundles in dicotyledonous root. In the previous session, we spoke about the different types of ways in which vascular bundles can be arranged in plants, right? We spoke about three different types: radial, conjoint open, conjoint closed. What we see in roots of both dicot and monocot is radial arrangement of vascular bundles, where the xylem and phloem are not present together in the same bundle. They are present separately in alternating radii, as you can see in this diagram. So what I'm going to highlight here, this is our xylem. And here in between that, you will have phloem. So if you can think of this as a circle, in one radius you have phloem, in the alternate radius you have xylem, again you have phloem, again you have xylem. So this type of arrangement of xylem and phloem is called radial arrangement. So in roots, both dicot as well as monocot, we can see radial arrangement of vascular bundles. Now when you talk about the number of xylem and phloem patches that are present in the root, usually in dicots, it will be in the range of 2 to 3. If there are 2, we call it diarch. If there is 4, we call it tetrarch. Okay, so here there are 4, so it is tetrarch. Diarch to tetrarch, it will vary. Now, when you consider the primary xylem here, remember in the previous class we learnt about endarch and exarch condition of xylem, right? So over here, you have to check for the position of protoxylem. Remember I told you that protoxylem vessels have narrower lumens, whereas metaxylem vessels will have broader lumens. So if you consider that, you can see that the protoxylem is present here and metaxylem is present here. Since protoxylem is present towards the periphery, such a xylem is said to be exarch. So over here in roots, you can see exarch condition of xylem, where the protoxylem faces the periphery and the metaxylem faces the pith. Okay. Now, conjunctive tissue. What is conjunctive tissue? Conjunctive tissue also is parenchymatous. So in between the xylem and phloem, whatever tissues are present between xylem and phloem, those are referred to as conjunctive tissue. All right. So following that, we have to learn about the pith. Pith is the central region which is also made up of parenchymatous cells. Pith in dicot roots compared with monocot's roots is inconspicuous. Inconspicuous means it is small, not very visible, not very evident. Okay. And like I already mentioned, this is parenchymatous. Now let's quickly summarize everything we've learned with respect to the dicot root. Outermost layer is the epidermis. Epidermis is single-layered parenchymatous barrel-shaped cells without any intercellular spaces. And there is no cuticle. There are no stomata. Some of the cells in the epidermis of root, which is also called epiblemma, give out extensions that are the root hair. Root hair are unicellular. They help in increasing the surface area available for absorption. Following the epidermis, you have the cortical region. Cortical region has several layers of parenchymatous cells that are kind of loosely arranged with a few intercellular spaces. These parenchyma cells have thin walls. Innermost layer of the cortex is called the endodermis. Endodermis is a single layer of parenchyma cells that are barrel-shaped, closely arranged without any intercellular spaces. The characteristic feature of endodermis in roots is that they show the presence of Casparian strips. Now, what are these Casparian strips? Casparian strips is the deposition of suberin in the radial and tangential walls of the endodermal cells. This suberin is impervious to water. It will not allow water to pass through it, and it's waxy in nature. Following the endodermis, you have the pericycle. Pericycle again consists of parenchymatous cells that are thick-walled. It may be multiple layers in thickness. And the pericycle is responsible for the initiation of lateral roots as well as for the initiation of vascular cambium during secondary growth. Now, talking about vascular bundles are radially arranged. Xylem and phloem alternate with each other and form patches. The number of patches can be 2 to 4. If you consider the xylem, since protoxylem faces outside, metaxylem faces inside, it is called exarch arrangement. The tissue, parenchymatous tissue that is present between the xylem and phloem patches is called the conjunctive tissue. The central tissue that is present in the center of the root, which is parenchymatous, is called the pith. There's another term that I want you to know about, which is stele. All of the tissues, including pericycle and everything else inside it, is called the stele. So pericycle, xylem, phloem, conjunctive tissue, pith, All of this is referred to as the stele. Stele here is S-T-E-L-E, not S-T-E-E-L. Okay. So stele includes the pericycle and everything else that lies within the pericycle. Alright, so that's about the TS of a dicotyledonous root. You can see that except for xylem and phloem, no other tissue is found here. Everything else is parenchymatous. From the epidermis to the pith, except for xylem and phloem, everything else is parenchymatous. Alright, now let's talk about the TS of a monocotyledonous root. TS over here again means transverse section. So when you take an extremely thin slice of a monocot root, put it under the microscope and observe it, here's what its section would look like. Again, this is only a portion of the section that we get, but this is just enough for us to understand what are the different tissues that are present in monocot roots. So the TS of monocot root and dicot root are not very different from each other. They're pretty much the same. There are only two major differences that we can find. One is with respect to the number of xylem and phloem patches. The other difference is with respect to the size of the pith. Okay. So as you can see, previously we had up to a maximum of 4 xylem and phloem patches. Here you have greater than 6. Okay, so it's called polyarch. Poly means many. Since you have many vascular bundles, it's called polyarch. Here you can see there is 1, 2, 3, 4, 5, 6, 7, 8. 8 patches of xylem and phloem. So this is one difference. The other difference is that the pith in monocot roots is more conspicuous than the pith in dicot root. So these are the only two differences that you can find between a monocot root and a dicot root. Otherwise, epidermis, cortex, endodermis, pericycle, conjunctive tissue, everything else is the same. It's only in the number of xylem and phloem patches and how conspicuous the pith is. These are the only two differences between a dicot root and a monocot root. Alright, now let's move on and talk about the TS of a dicot stem. So again, here, if this is a plant, a dicotyledonous plant that has leaves, root, you're taking an extreme thin slice over here. Then you take a section of a somewhat cylindrical stem. Then the section is going to appear circular in outline, which is what we get over here. And when you take just one portion of this, like this, here's what you get. We're focusing only on one region of the section. Whatever we learn about here applies to the whole of the root. The stem. Alright, so this is what a dicotyledonous stem section would look like. I want you to notice one thing here. Look at how all the vascular bundles are arranged in the form of a ring. All these triangular structures that you're looking at here, these are nothing but the vascular bundles, that is xylem and phloem. See how neatly they are arranged in the form of a ring. Right? So that is the TS of a dicot stem. Now let's focus on just this one diagram and learn about the different types of tissues that we can find there. I've already mentioned it multiple times before. The outermost layer of any part of the plant that you take is going to be the epidermis. So is the case for dicot stem. Epidermis is the outermost layer. Since this is an above ground part of the stem or an aerial part of the stem, this is covered with a thin layer of cuticle. Some plants, young stems, they bear stomata, and some of them may even bear hair on their surface, which we call trichomes. Okay, the difference between hair that is found on the surface of roots and stem is that in the root, like I mentioned earlier, unicellular root hair are found, but the hair or trichomes that you find in dicot stems, these are multicellular in nature. Okay, in order to represent that, you have these lines here. See in the illustration. So these septa or lines here represent that the trichomes are generally multicellular. Okay, so that's about the epidermis. Now talking about the cortex. In the cortex, you can find 3 distinct zones in case of dicot stem. So it's a little complex cortex that we see here. The region of the cortex immediately below the epidermis is called the hypodermis. Now what does hypo mean? Hypo means below. What does epi mean? Epi means above. So just below the epidermis, the zone of cells that you find in the cortex, that is called as the hypodermis. Now this hypodermis is a few layers in thickness that is represented by these pink colored cells here. This hypodermis is colon-chymatous in nature. Remember when we spoke about colon-chyma in the previous lecture, I told you that it is found only in dicots and mostly you find it in the dicot stems just below the epidermis in the hypodermis. So this is exactly where you can find colon-chyma tissue. You find this in young stems and this provides mechanical support and a little bit of flexibility to the plant. Following this, you have the general cortex. The general cortex here is very similar to the cortex that we found in the section of roots. So it is parenchymatous, it is kind of relatively loosely arranged and it has intercellular spaces. So it is rounded, thin-walled parenchymatous cells with conspicuous intercellular spaces. Endodermis is the innermost layer of the cortex. Endodermis here is also made up of parenchyma cells and it is rich in starch. Since it is rich in starch grains and it has starch grains, it is known as starch sheath. The thing about endodermis and roots that you have to remember is about Casparian strips. The thing in dicot stem about endodermis that you have to remember about is the starch sheath. Endodermal cells in dicot stem possess starch grains, which is why it is also called the starch sheath. All right, so that's about the cortex. So colon-chyma you have in the hypodermis, parenchyma general cortex, endodermis also is parenchymatous in nature. All right, now let's move on and talk about the pericycle. The pericycle in case of dicot stem is made up of sclerenchyma tissue. The thing is, it is present in the form of semilunar patches. So if you take this as the epidermis and this as the endodermis, I told you how the vascular bundles are arranged in the form of a ring, right? So the vascular bundles will be arranged like this. So towards the inner side of the vascular bundle, there's xylem, 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 xylem. Outer side you will have phloem, 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 phloem. Okay. Now pericycle is present on top of this like this. Okay, so this region is the pericycle. As you can clearly see, it is semicircular, right? What does semilunar mean? Semilunar means half moon shaped or semi, see, like this. So this is present in the form of semilunar patches. It is present inside. This red layer is the endodermis. It is present inside the endodermis and above the phloem. That is exactly the location of sclerenchymatous pericycle here. All right. Now let's move on and talk about the medullary rays. Now what are these medullary rays? Medullary rays are the parenchyma cells that are present between the vascular bundles. Over here between the vascular bundles you will have parenchyma cells here wherever I'm highlighting. These are called medullary rays. Why do we call them medullary rays? Because they are radiating from the center, right? How do you draw sun rays? Sun rays you draw like this. Similar to sun rays, you have parenchymatous cells that are radiating from the central pith. That is why we call it the medullary rays. Okay, now let's talk about, again these are also parenchymatous. Now let's move on and talk about vascular bundles. Vascular bundles in dicot stem is conjoined and open, which means they are found in the same bundle. Within the same bundle you have, let me use dark blue here. So this region you have the phloem. Here you have the xylem. In between them, you have the cambium, right? So the xylem and phloem here are present within a single bundle. Therefore, it is conjoined. Open because there is a strip of cambium that is present between the xylem and phloem. Okay, so they are arranged in the form of a ring, which we clearly know. I showed it to you earlier also. It's arranged in the form of a ring. And it is conjoined because they both xylem and phloem are present in the same bundle, unlike in roots. And it is open because there is a strip of cambium that is present between the xylem and phloem. Over here, we see endark condition of xylem. If it has to be endark, the protoxylem has to face the inside, metaxylem has to face the periphery. Over here, the protoxylem with the smaller lumen, it faces the pith or the inside, while the metaxylem, which has the wider lumen, faces the outside or the periphery. So clearly over here we can see endark arrangement of primary xylem. Now let's talk about the pith. Pith, as usual, is parenchymatous here. You can find rounded cells with intercellular spaces in the pith, okay? And the pith is conspicuous. It's very prominent. That's about the TS of a dicot stem. Let's quickly summarize before we move on to the TS of monocot stem. Epidermis, single layer, parenchymatous, trichomes may be present, stomata may be present, cuticle will be present. Below that you have the cortex. Cortex has 3 zones. Just below the epidermis, multi-layered hypodermis, which is collenchymatous. Below that you have general cortex, which has parenchyma with intercellular spaces. Innermost layer of the cortex is the endodermis, which also has parenchymatous cells. In the endodermis, you can see that the cells store starch grains, which is why it is called the starch sheath. Now, inside the endodermis, you have pericycle. Pericycle is arranged in the form of semilunar patches. It is composed of sclerenchymatous tissue. And in the vascular bundles, talking about the vascular bundles are found as conjoint open type of vascular bundles where xylem and phloem are together, and in between them there is a strip of cambium. Therefore, it is conjoint open type of vascular bundle. In between the vascular bundles, you have parenchyma tissue radiating from the center, which we call medullary rays, and in the center of the stem, you can find parenchymatous tissue forming the pith. So it is in dicot stem that you can find all the types of tissues that you've studied about. We saw about a lot of parenchyma in the cortex, in the endodermis, epidermis. We spoke about collenchyma in the hypodermis. There's sclerenchyma in the pericycle. There is xylem and phloem vascular bundles. So every type of tissue, permanent tissue that you've studied about, you can find in this section of the dicot stem. Alright, now let's move on and talk about the monocot stem. Monocot stem, if you take a section, it will appear circular like this. As you can see here, the vascular bundles are scattered, right? So that is the characteristic feature of monocot stems. The vascular bundles will be scattered here. Now let's focus on just this one portion of the TS of monocot stem. As always, the outermost layer is the epidermis. Just below the epidermis, you have hypodermis, which is made up of sclerenchyma. Both dicot stem and monocot stem, both of them have hypodermis, distinct hypodermis. The hypodermis in dicot stem is collenchymatous. The hypodermis in monocot stem is sclerenchymatous. Okay. So epidermis is parenchymatous, single layered. Hypodermis is sclerenchymatous. And below that, you have ground tissue. You do not see any differentiation between endodermis, pericycle, none of that you will see. There is ground tissue, and interspersed within this ground tissue are the vascular bundles. So you can see how the vascular bundles are scattered here. See, these are the vascular bundles that are scattered within a homogeneous ground tissue. These vascular bundles are conjoint and closed, which means it is present in the same bundle, xylem and phloem, but there is no cambium present between the xylem and phloem. Okay, and these vascular bundles are surrounded by sclerenchyma cells. Because it surrounds the vascular bundles and forms a sheath, we call it bundle sheath. Vascular bundle is being covered, right? That is why we call it the bundle sheath. Now, if you look at the size of vascular bundles, you can see how it's variable. The ones that are present towards the periphery are smaller in size, while the ones that are present towards the center are comparatively larger in size. So that difference in size can be observed in the TS of a monocot stem. Okay, so the peripheral ones are smaller than the centrally located ones, which are relatively larger. Sometimes the vascular bundles contain water-containing cavities. Phloem parenchyma is absent in monocot stem. When I discussed phloem also, I told you that parenchyma is usually absent in monocots. It is mostly found in dicots, right? So the same thing is repeated here. So that's about the TS of a monocot stem. You have the epidermis, which is the outermost layer, hypodermis, sclerenchymatous. You have a ground tissue, no distinction between endodermis, pericycle and all of that. Within the ground tissue, vascular bundles are scattered. The size of the vascular bundles towards the periphery are smaller compared to the ones that are present at the center. The vascular bundles are conjoined and closed. The present xylem and phloem is present in the same bundle, but there is no cambium present in between them. All right, these vascular bundles are surrounded by sclerenchymatous tissue, which we call the bundle sheath. All right. Now let's talk about the TS of a dicot leaf. Dicot leaf is also called dorsiventral leaf. Why do we call it dorsiventral leaf? What is dorsal side? For example, let's say this is a book. This is your NCERT textbook. So if you can think of this as a leaf, if you take a section like this, if you cut a very thin slice of the leaf like this and observe it, it's going to appear like this. Okay, so this circle that you're seeing here is the vascular bundle that represents the vein in the leaf. This is the upper epidermis. This is the lower epidermis. There is distinction between the upper surface and the lower surface in dicot leaves. That is why we call it dorsiventral leaf. Dorsal side and ventral side are not identical. There are some differences we can see in the dorsal side and the ventral side of the leaf. Therefore, it is called a dorsiventral leaf. Okay, let's talk about it. Epidermis. You can see epidermis on both sides of the leaf. The epidermal surface on the upper side is called the adaxial epidermis. D is direct. Okay, so if you get confused between the terminologies, you can remember this. Adaxial is the upper surface. You can directly see the upper surface of the leaves. Abaxial, B for below. Abaxial epidermis is the lower surface of the leaves. Okay, so you have the upper epidermis, lower epidermis, adaxial epidermis it is also called. This is called abaxial epidermis. In leaves, we find the presence of a very thick conspicuous cuticle. Cuticle is waxy in nature. It prevents the loss of water by transpiration. Since maximum transpiration happens through leaves, a very thick cuticle can be seen on the surface of leaves, which is represented here on the upper surface and the lower surface, both surfaces. On the outer surface of the epidermis, you can find cuticle deposition. Okay, now, yeah, talking about the stomata, you know that epidermis is also associated with stomata, right? The number of stomata is variable over here. On the abaxial epidermis, stomata number is high. On the adaxial epidermis, stomata number is low. Sometimes it may even be absent. So, this is the difference between the upper surface and lower surface with respect to the epidermis. Now, when we talk about the cortex, the ground tissue in leaves is called the mesophyll. Phyll itself means leaf. Meso means in between. So the tissue that is present between the upper and lower epidermis is what we call the mesophyll. Okay, so the ground tissue is called mesophyll in leaves. This I told you earlier also in the previous class. I hope you remember that. Now in the mesophyll we have chlorenchymatous tissue, parenchyma containing chloroplast. Because leaf is the site of photosynthesis, photosynthesis has to happen, pigments are required. Pigments are present in the thylakoids, present in the chloroplast. So wherever photosynthesis needs to occur, over there the cells should have a large amount of chloroplast in them. Okay, so these mesophyll cells have a lot of chloroplasts in them. So within the mesophyll itself, in dicot leaves, you can find two types of cells. One type of cells are kind of elongated. They are arranged without many intercellular spaces. They are arranged parallel to each other like this. This type of parenchyma is called Pericycle parenchyma. It is present towards the upper epidermis. That's why it's said to be adaxially placed. They are elongated cells, like I already said. They are arranged vertically parallel to each other. Now, the second type is called the spongy parenchyma, which I'm going to highlight in dark blue color over here. These spongy parenchyma are somewhat irregular shaped or sometimes may even be spherical. You can see how they are very loosely arranged, and there are a lot of air cavities between the cells here. Air spaces are present. So this is abaxially placed, oval or round, very loosely arranged. Many large spaces or air cavities are present here. Now over here, the cells are compactly arranged without any intercellular spaces in Pericycle parenchyma, and the cells will have a lot of chloroplast, whereas in abaxial epidermis towards the lower epidermis, we have spongy parenchyma which are loosely arranged with a lot of intercellular spaces. Because of the compactly arranged nature of Pericycle parenchyma and because of the high number of chloroplasts that you can find in them, usually when you look at the leaves, the upper surface is going to be relatively more darker compared to the lower surface. Usually upper surface will be dark green and lower surface will be slightly lighter in color in dicot plants, right? So that is because of this kind of arrangement of parenchyma. That's about the mesophyll. Now let's talk about the vascular bundles. Vascular bundle size varies in dicot leaves because all of the veins in the leaf are not of equal size. The thickest vein is usually the midrib. Thinner than that is the next branch. As it goes on branching and forming a network, the diameter of the veins will reduce. Because it is within the veins that vascular bundles are present, when you take a section, you can see that the vascular bundles also vary in size. The central midrib will be thickest, will have the widest diameter. Following that, you will find vascular bundles that are relatively smaller. Okay, so this is seen in the midrib and in the veins. The size of the vascular bundle depends on the size of the veins. Usually, it is surrounded by a layer of thick-walled bundle sheath cells. Bundle sheath because it is covering the vascular bundles. So that's about dicot leaf or dorsiventral leaf. Adaxial epidermis, abaxial epidermis, both are there. Covered by cuticle, you have stomata in the epidermis, more in the lower surface compared to the upper surface. Upper surface very few or sometimes even absent. What lies in between these two is the mesophyll tissue. In mesophyll, you find two different types of parenchyma, palisade and spongy. Palisade, elongated, rod-shaped, vertically arranged, parallel to each other, contain a lot of chloroplasts. Towards the abaxial surface, you have spongy parenchyma, which are somewhat oval to polygonal, loosely arranged with a lot of intercellular spaces. And then we have the vascular bundles present in the regions of the veins and the midrib. Size is variable according to which vein it is present in. Surrounding the vascular bundles, you have a layer of cells that is called the bundle sheath, which has thick walls. Now let's talk about a section of isobilateral leaf. Isobilateral leaves are nothing but dorsiventral leaf. Iso means same or similar. I've already told you that. Bilateral means two sides. Lateral means side. Isobilateral means both the sides of the leaf are pretty much similar. Okay. Now in isobilateral leaves, you find the epidermis. Epidermis, on the outer surface, you will obviously find the cuticle. Over here, the stomata are present on both surfaces and they are pretty much arranged equally in number. They are pretty much equal in number on both surfaces, upper surface as well as lower surface. And in grasses, which is a type of monocot, certain adaxial epidermal cells along the veins. If this is a vein, if this is the vascular bundle, certain cells along the vein, they become modified. They become very large and empty. Compared to the other cells, they become larger in size, they become empty and they become colorless. Such cells are called bulliform cells. This is one important thing that you will have to remember with respect to the section of an isobilateral leaf. Now, what is the function of these bulliform cells? Whenever these bulliform cells are turgid, whenever there is a lot of water inside these bulliform cells and they're very turgid in nature, at that time the leaf surface is exposed. Now let's assume the plant is under water stress. There's not sufficient water present in the soil for the plant to absorb. So the transpiration rate, if it increases, photosynthesis cannot happen very well. In such cases, the plant has to stop water loss from it so that it doesn't dry to death. Okay? So what it does in such cases is, in order to reduce transpiration, it will lose water, it will become flaccid. Once these bulliform cells become flaccid, the leaves will roll inward. As the leaves roll inward, the exposed area to the sun or to the outer atmosphere will reduce. Therefore, by that way, water loss by transpiration can be reduced. Okay, so when it is turgid, leaf surface will be exposed. When it's flaccid, leaves curl inward in order to minimize water loss. Now let's talk about mesophyll. Here you do not see any differentiation into palisade and spongy. All of them appear the same kind. They have a lot of chloroplasts. They are parenchymatous in nature, and it's not differentiated into spongy and palisade parenchyma. Talking about vascular bundles, since it is parallel venation that you see in monocots, except for the main midvein, every other vein will be nearly similar in size. So the size of the vascular bundles in the section also pretty much the same. Okay. So that's about the section of isobilateral leaf. Adaxial epidermis, abaxial epidermis, both are similar. Both of them possess cuticle. Both of them have stomata in somewhat equal numbers. Within that you will find parenchyma, mesophyll. All of them are similar, no distinction between palisade and spongy. They all have chloroplasts, perform photosynthesis. Vascular bundles, the size of the vascular bundles are pretty much the same because it is parallel venation, right? So this is the summary of isobilateral leaf. Now let's talk about secondary growth. Now what do we mean by secondary growth? Secondary growth basically is increase in the girth. Now let's take an example of a mango sapling. So when you sow a mango seed, what happens? The seed germinates, it forms a plant. It forms a plant, right? Now this plant will have initially a very thin herbaceous stem, and then it grows into a short plant. Over the course of time, if this plant has to become like a mango tree, then two things are happening. It is increasing in height or length, and the stem is also increasing in girth. The growth that happens where there is an increase in length is called the primary growth. The growth that happens because of which there is an increase in girth is called the secondary growth. The reason for primary growth or increase in length to take place is because of the apical meristems that are present at the root tip and the shoot tip of plants. Okay, so we've already covered that in the previous lecture, so I will not get into the details of it. Apical meristems are responsible for increase in the length of the plant, whereas lateral meristems are responsible for increase in the girth of the plant. Now, when you consider secondary growth, this is exclusive to dicot plants. It doesn't happen in monocots. It happens in dicots. It also happens in gymnosperms. But since this chapter is about flowering plants or angiosperms, we will not focus on gymnosperms. We learn about how exactly the secondary growth will take place in dicot plants. So now for that, I want you to recollect the section of dicot stem as well as dicot root as we go forward. But for now, I want to tell you that this secondary growth in the stem occurs in two areas. So if you take this as the section of a dicot stem, wait, I think I have an illustration here. So if you can think of this as a section of a dicot stem, over here cambium is present. Cambium is present within the stele. Because of the activity of this cambium, secondary growth will take place. Also, a cambium will form in the cortex region, because of the activity of that also increase in girth or secondary growth will take place. The cambium that is present within the stele is called the vascular cambium, and the cambium that is present in the cortex is called the cork cambium. So over here I said that primary growth happens because of apical meristem, secondary growth happens because of lateral meristem. In lateral meristem you have vascular cambium and cork cambium. Vascular cambium is present within the stele inside. Cork cambium is present in the cortex region. The cells in both these cambia or both of these meristematic tissues will divide and help in increasing the girth of the plant. Okay. Now, secondary growth is something that you have not learnt before, right? You're learning it for the first time. So it's very important for you to pay attention and listen to it with a focused mind so that you understand this very well. Otherwise, if you want to or even try to study this by your own, you may not be able to understand it very well. This is a very important concept for your competitive exams as well as for your board exams. So I request you all, I urge you all to pay undivided attention to what I'm going to say for the next couple of minutes about secondary growth. Okay, it's interesting and it is important. All right, now we already learned about the section of Dicot Stem, right? In today's lecture, I told you about the section of Dicot Stem. I want you to recollect what we learnt about the vascular bundles there. I told you that the vascular bundles were conjoined and open, right? So this is representing the central portion of the TS of a dicot stem. First, we'll learn about how secondary growth happens in the dicot stem. Then we'll move on to learn about the secondary growth in dicot root. All right. So over here, we have vascular bundles arranged in the form of a ring. Within each vascular bundle, you have xylem towards the center. I'll use a different color. So we have xylem here throughout. Basically, this is primary xylem that was present since the beginning of the life of the plant. On the outside, we have phloem. In between the xylem and phloem, we have a strip of lateral meristem which we call the cambium. Now let's give this cambium a name. We will call it vascular fascicular cambium or intra fascicular cambium. Okay, so this is called intrafascicular cambium. Now what does the word intra mean? Intra means within. This cambium is present within the vascular bundle between the xylem and phloem. That is why we call it intrafascicular cambium. All right. Now, we know that in between the vascular bundles, whatever parenchyma is present here, we call it the medullary rays. And these medullary rays are parenchymatous in nature. So if it is parenchymatous, that means it is a permanent tissue. Now, some of these cells of the medullary rays adjoining the cambium or at the level of the cambium will become de-differentiated. Remember what the difference between meristematic and permanent tissues is? Meristematic tissues have the ability to divide. Permanent tissues do not have the ability to divide. Now this green colored cambium that is present between xylem and phloem is meristematic in nature. The cells that are present there have the ability to divide. Now the medullary rays that are present between the xylem and phloem is made up of parenchyma. This has lost the ability to divide and become permanent. Now some of these parenchymatous cells that are present in the medullary rays adjoining the vascular cambium, adjoining the intravascular cambium at the same level of the cambium, what happens here is they are initially permanent, lacking the ability to undergo cell division. They regain the ability to undergo cell division and become meristematic again. Okay, so this process is called de-differentiation. I gave you an example and explained to you what exactly it is in the previous class. De-differentiation. So this, whatever in dark blue I have drawn, in that zone, in the medullary rays, the cells that were parenchymatous and permanent and lost the ability to divide, they regain the ability to divide and become meristematic. Now those meristems are called interfascicular meristem. Whatever is in green is intra because it is within the vascular bundles. Whatever has formed by de-differentiation now, secondary meristem that is formed now, that is interfascicular meristem. Inter means in between. Since they are present between the vascular bundles, it is called interfascicular cambium. Now this interfascicular cambium and intrafascicular cambium combine together to form a single continuous ring like you can see here. Okay, so both of these combined to form a ring which we call the cambial ring. To be more specific, let's call it the vascular cambial ring. Understood? Intrafascicular cambium present within the vascular bundles, join with the interfascicular cambium which is present in between the vascular bundles to form a continuous ring called vascular cambium. Okay, that is the formation of the cambial ring. Now what happens is, let's say this just give me a minute. Okay, so let's say this is the vascular cambial ring. Okay, now this vascular cambial ring will have, let's say these are the vascular bundles. So there's xylem inside, phloem outside, xylem inside, phloem outside. Okay, so this vascular bundle will have this cambial ring. The cambial ring will have somewhat rectangular cells present in it. Okay, so it's a thin strip of cells present like this. Throughout the cambium, you will have cells like this. Now what I will do is I will focus on this region alone. So here you have rectangular cells present in the cambium. Remember, these cambium, the cells in the cambium, they are meristematic. They can divide. Apart from these cambial cells, the cells in the pith, the xylem, phloem, none of them are capable of undergoing cell division. Okay, so if this stem has to increase in girth, more cells have to be added. So where are those cells that have the ability to divide present? They are present in the cambium. So what exactly is this cambium going to do now? It is going to undergo cell division in this plane. So if this cell here divides, it adds a cell inside and there's a cell outside. If this cell divides, a cell is formed inside, cell is formed outside. Like that, this cambium will go on undergoing cell division and cut off cells towards the inside as well as towards the outside. Whatever cells it cuts off towards the inside will form secondary xylem. Whatever cells it cuts off towards the outside, that will form secondary phloem. As the plant is growing in height, as the plant is growing in general, more water will be needed for the plant. If more water has to be needed for the plant, more xylem elements have to be added to the plant. So in order for adding more xylem elements, the cambium is helping. As the cambium will add more and more xylem and phloem elements, the stem will naturally increase in girth, right? So this cambium will undergo divisions in this plane. It will cut off cells towards the inside over here, and it will also cut off cells towards the outside. Whatever cells it cuts off towards the pith will become the secondary xylem. Whatever cells it cuts off towards the outside will become the secondary phloem. So whatever xylem and phloem you have here, this is the primary xylem, this is the primary xylem, this is the primary phloem. These were present in the plant from the beginning. Now, because of the activity of cambium, whatever xylem and phloem are newly forming later in the life of the plant, they are called secondary xylem and secondary phloem, right? So you can see that towards inside secondary xylem, towards outside secondary phloem is adding. Sometimes in between it will add narrow strips of parenchyma both inside and outside. That is referred to as secondary medullary rays. Now this cambium is cutting off cells inside and outside. Whatever cells it's cutting off inside will become xylem, we call that secondary xylem. Whatever cells it's cutting off towards the outside becomes phloem, we call it the secondary phloem. In between here and there, in between the secondary xylem and phloem, narrow strips of parenchyma is being added on both sides. We call that secondary medullary rays. Okay. Now, when you talk about the activity of this cambium, it is more active in cutting off cells and forming secondary xylem compared to secondary phloem. So, the number of secondary xylem will be greater than secondary phloem. The cambium produces more xylem compared to the secondary phloem. As more and more xylem gets accumulated in the center and more material is added towards the center, the primary xylem and primary phloem that are present towards the outside will start getting crushed. Okay, so that is what will happen during secondary growth. Now, talking about the activity of the cambial ring. The cambial ring's activity varies seasonally. According to the season, according to the external environment, the cambial activity will vary. Whenever in trees that are found in temperate regions, you know how the seasons will vary drastically, right? There is an extreme cold season, there is an extreme hot season. So seasonal variations are very distinct when it comes to temperate regions. So since the activity of cambium is under the control of so many physiological as well as environmental factors, you do not see the same kind of cambial activity throughout the year. In temperate regions, climatic conditions, like I already mentioned, are not uniform. They are not pleasant or they are not the same throughout the year. It varies from season to season. The cambial activity during spring is very high compared to autumn. During autumn, it is low. Because of this variation in the activity of cambium, you can find that two types of secondary xylem vessels are produced. The activity of cambium is very high, very active during spring. So during spring, whatever vessels are produced, their number is more and the lumen's diameter also is more. So more number of xylem vessels are produced during spring. The width of the xylem vessels is also greater in the vessels that are produced during spring. So whichever xylem is formed during spring, that is called spring wood or early wood. Clearly it is formed during spring. The number of xylem elements is large and the vessels also have a wider cavity. Coming to autumn wood, during autumn, the activity of cambium is not very great. So the number of xylem or xylem elements that is formed during autumn is less, or xylary elements is less. And also they have a relatively narrower cavity. The spring wood is that is formed during spring has low density and it is light in color, whereas autumn wood has higher density and it is darker in color. In temperate plants, in temperate trees, this can be distinctly seen when you take a section of the tree trunk. You may have heard how you can count the age of a tree by counting the number of rings. So this is exactly how it happens. They are called annual rings. Annual means what? Yearly. So in a year, how many times do we get spring and autumn? You have one spring and one autumn in a year. So in a year, at one time, more number of xylary vessels or xylary elements with wide lumens are produced. In the same year, number of xylem vessels is less and narrower, that is produced during autumn, right? So in a year, there are two bands that you can see being formed. Okay, so let me show it to you here. Over here, if you look at this here, from this dark band to, let's say, this dark band, okay? So over here, you can see a light region, right? Wait, I'll use this color. So whichever region I'm highlighting here, this region is light in color. This represents spring wood. This region is dark in color. This represents autumn wood or late wood. In a year, you will have one light colored ring formed because of spring wood, one dark because of autumn wood. So one spring wood and one ring of spring wood and one ring of dark wood or autumn wood, this will give us one annual ring. Okay, so spring wood and autumn wood that appear as alternate concentric rings. You can see how it alternates. First you have a dark autumn wood, then you have light spring wood, then you have dark autumn wood, light spring wood, dark autumn wood, light spring wood. So that alternates season after season. In a year you can find two rings. It is because they are so distinct with respect to their color and density, you can see them as rings. If cambial activity is uniform throughout the year, you cannot find such rings. Therefore, such rings are not very evident in all kinds of trees. In some places in the world, the external or environmental conditions are pretty much uniform throughout the year. Over there, you do not find such rings. So these rings can only be found in regions where there is seasonal variation in temperature and other environmental factors. Okay. So whenever that is the case, the spring would be lighter in color and it will be wider. The autumn wood will be darker in color and it will be narrower. One spring wood and one autumn wood together will give us one ring of spring wood and one ring of autumn wood would give us one annual ring. Okay, so this constitutes one annual ring. We can use this to estimate approximately what could be the age of the tree, because for every year this growth is happening, you're finding two concentric circles of spring wood and autumn wood, so that would count as 1 year. So by that way, if you count, you can kind of estimate the age of the tree, and that is called dendrochronology. Okay. Now, let's talk about heartwood and sapwood. Whenever you've looked at logs of wood, you may have noticed that sometimes the central region of the log of wood is darker in comparison with the outer lighter region. So, this is because the center region, which is called the heartwood. Okay, let me just tell you what it is called first. The central dark region is called the heartwood. The surrounding lighter region is called the sapwood. Heartwood and sapwood are different from each other in a certain things. I'll tell you what they are. Heartwood is the innermost layer of the stem. It is dark brown in color, as you can clearly see. The reason it is dark in color is because it has many things deposited within it, such as tannins, resins, oils, gums, aromatic substances, essential oils. All of that is deposited here. Because it has the deposition of all of these substances, that makes this portion of the wood, which is heartwood, durable and resistant to the attack of microbes and insects. Okay, so this region called the heartwood is dark in color. It has many substances deposited within it, which is what is responsible for its color. And also because of the deposition of those substances like tannins, gums, that makes this part of the stem very durable. So when they make furniture out of wood, it is mostly this wood that they prefer because it is not prone to insect attack. Okay, so this makes it durable. And within this, whatever xylem elements are present, whatever conducting vascular bundles are present, they do not function. Okay, so they are dead elements with highly lignified walls. The xylem elements that are present within this, they are dead. They do not perform their function. Their walls are very lignified. They do not conduct water. Then what is the purpose of it? I told you that xylem has two functions. One is conduction of water. The second thing is that it provides mechanical support. This xylem in the heartwood does not conduct water, but it provides mechanical support to the plant. Now let's talk about sapwood. It is the peripheral region. Clearly it is much lighter than the heartwood. And the xylem that is present in the sapwood conducts water for the plant. Okay, so that's the difference between your heartwood and sapwood. Now, we finished learning about how secondary growth takes place because of the vascular cambium. Now we have to learn about how secondary growth takes place because of the cork cambium present in the cortex. If you remember, when we talk about the dicot stem, let's say this, yeah. So this is the epidermis. Let's say this is the vascular cambium. Okay. Okay, so you have vascular bundles here. I'm only drawing what is relevant to us right now. So if you think this is the epidermis, this is the vascular cambium, this is xylem and phloem. Vascular cambium is undergoing cell division, producing secondary xylem, secondary phloem. That is happening. Now, as here more and more xylem elements are getting added, there is pressure on the outer side. The epidermis could start cracking because of the pressure that is developing on the inside. Epidermis's function is to protect whatever is lying inside the plant. So over here what can happen is the epidermis can rupture because of the pressure that is being generated towards the inner side of the plant because of activity of the cambium. Therefore, newer protective layers have to be formed outside to protect the plant. Okay, so what happens there is, from the cortex, a strip of cells from the cortex, let me use yellow. Okay, so these are, if you can think of this as the cortex, it has parenchyma cells. This cortex will undergo dedifferentiation. It will become meristematic again. And form the cork cambium. The cork cambium has another name which is called phelogen. Similar to the vascular cambium, the cells in the cork cambium also will undergo cell division and cut off cells towards the inside and outside. So whatever cells are cut off from the cork cambium or phelogen towards the inside is called phylloderm or secondary cortex. Whatever it cuts off towards the outside is called phylum or cork. So this is the cambium, cork cambium to be more specific, or phyllogen. Okay, so these three layers, phylloderm, cork cambium, phylum, together are called, where do I write it? All of these together, phylum, cork cambium, phylloderm, together is called the periderm. Now, these phylum cells or cork cells that are cut off to the outside will have suberin in their walls. They will be impervious to water. It will not allow water to pass through it. Okay, so that is the cork cells. Whatever is produced in the phylloderm, the secondary cortex, these are parenchymatous cells very similar to the general cortex. Okay, now sometimes the cork cambium, instead of producing cork cells towards the outside, will produce compactly arranged parenchyma cells. Over here, look here. I'm going to show you a section of cork cambium here. Whatever I'm highlighting here is cork cambium. Towards the outside of cork cambium, you have phylum or cork. Towards the inside, you have phylloderm or secondary cortex. Okay, so normally cambium is supposed to give cork cells outside, secondary cortex, so periderm inside, right? I'm sorry, phylloderm inside. But sometimes what the cork cambium does in certain locations is, instead of producing phylum or cork cells towards the outside, it will produce compactly arranged parenchyma cells instead. Compactly or closely arranged parenchyma cells. Now these parenchyma cells will rupture the epidermis and create lens-shaped openings here in the epidermis. Those lens-shaped openings are called lenticels. So these lenticels will help provide an opening for the exchange of gases and for transpiration. Okay, so that is about the lenticels. I hope you've understood what I explained here about the cork cambium. Cork cambium is entirely produced by dedifferentiation of parenchyma in the cortex. It will divide and cut off cells towards the inside as well as outside. Whatever cells it cuts off inside, it is called the phylloderm or secondary cortex. It is parenchymatous in nature. Whatever it cuts off towards the outside, it is cork cells. It is also called phylum. So these cells are very thick walled. They have suberin in their wall and they are impervious to water. Sometimes the cork cambium, instead of producing cork cells towards the outside, it will produce closely arranged parenchymatous cells called complementary cells, which will rupture the epidermis. If the epidermis ruptures, small lens-shaped openings will be formed in the epidermis. They are called lenticels. These lenticels help in the exchange of gases and also for transpiration. Okay, so that's about the lenticel here. Now let's talk about the bark. I'm sure you all know what a bark is. It is a very common term that we use. So bark, the way we think about it in layman terms, it is the outermost hard layer of a tree trunk. But botanically, if you have to speak, bark comprises of all the layers that are present in a stem outside the vascular cambium. So you know vascular cambium is present. So all of the tissues that are present outside vascular cambium is the bark. Okay, so technically it includes secondary phloem, primary phloem, pericycle, cortex, epidermis, everything is covered under bark. or everything is included under bark. Now we've learnt about secondary growth in Dicot stem. We learned how it happens in the vascular cambium because of the vascular cambium. We also saw how it happens because of the cork cambium. Okay, in both cases we see that it accumulates or produces cells inside as well as outside, and as a result of that the girth of the stem will increase. Now it's time to learn about secondary growth in dicot roots. Remember, in dicot root there is no cambium that was present in the vascular bundles, right? So whatever cambium will form has to form entirely by dedifferentiation. Now let's look at the stele region of dicot root. Okay, so this is the stele region of dicot root. So you have xylem, 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 and then you have phloem here. A ring will form above the xylem near the protophlloem. Above the phloem near the protoxylem. Here above the phloem, pericycle near the protoxylem. A continuous wavy ring is going to form like this. See, this is our protoxylem. This is our phloem. So the cells that are present above the phloem and the cells, pericycle region near the protoxylem, they become dedifferentiated and they form a continuous wavy ring like this. Okay, so that is what you can see over here. This wavy ring will become meristematic and it will function very similar to the vascular cambium that we saw in the case of dicot stem. Similar to that, it will give rise to secondary xylem towards the inside. So where is the ring? Okay, so if you could think of this as the cambium, it will produce secondary xylem towards the inside, secondary phloem towards the outside. Okay, so very similar to how secondary growth occurred in the stem of a dicot plant. Even in the root, once the cambial ring forms because of dedifferentiation, it will cut off cells inside as well as outside. Whatever cells it cuts off inside form the secondary xylem, and whatever cells it cuts off towards the outside will form the secondary phloem. Okay, so that's about secondary growth in dicot roots. So with this, we have covered pretty much everything in this chapter. Today's session, we started off by learning about the different types of tissues that are present in monocot and dicot roots, stem and leaves. Following that, we learned about secondary growth. I understand that it's a slightly new concept for you, which can be a little, intimidating, or it can seem a little daunting for you. But trust me, once you understand this concept, you will enjoy it. So watch this lecture at your own pace, pause as many times as you want, but understand this concept fully because it's very, very important for your exams. All right. So until I see you in the next class, all of you all take care. Thank you.